Swayam Prabha. Digital India. Educated India.
Hello and welcome to this lecture on neoplasia. In this lecture, we will talk about the hallmarks of cancer, the fundamental properties of cancer, what are known as the hallmarks, the phenotypic and biological characteristics. And we will take up one of the main hallmarks of cancer, which is uncontrolled growth. And we will try to see what is the molecular mechanism behind this particular hallmark of cancer. Okay, this phrase, the hallmarks of cancer, was first used and published in the Cell by Weinberg and his colleagues in 2011, and they have got the attention of all the people who study cancer all over the world, because they sort of in a nutshell try to tell you what are the properties of a cancer cell. So, it is almost a complete definition of a cancer cell. So, let us go over each of these a little bit, and then we will go to the first one, the self-sufficient growth signaling, the most important aspect of a cancer cell, a little bit more in detail in the rest of the lecture. So, firstly, a cancer cell has self-sufficient growth signaling, which means it can it is self-sufficient, it does not need external agencies, it will just trigger its own growth signaling and go on proliferating. The second most important property of a cancer cell is its insensitivity to growth inhibitory signals. Normally, when cells divide, they stop at a certain point because they are sensitive to the inhibitory signals around them, but cancer cells do not listen to any of these signals, they continue to proliferate. The third important aspect of a cancer cell is there is an altered cellular metabolism. The cancer cell metabolism is not like a normal cellular metabolism, it's a completely different cellular metabolism. They evade apoptosis, they are resistant to cell death. So, normally a cell, when it is damaged or when its life span is over, it undergoes apoptosis and natural cell death, whereas a cancer cell evades apoptosis. In other words, it has, in that sense, it has certain immortality. It enables replicative immortality, which means it does not stop replication, it goes on multiplying immortally. There is sustained angiogenesis, which means in order for a tumor to grow, it needs nutrition, isn't it? So, it has a tumor ensures that it has its feeding vessels, and there are new blood vessels that sprout up and which carry nutrition to the tumor and enable it to grow indefinitely. And 7, its amazing quality to invade and metastasize. I think this is the one property of cancer that makes it extremely difficult to treat in a certain patient. And finally, it avoids immune destruction. So, there is a whole lot of work now going on tumor immunity. Why everyone does not develop cancer more frequently than what it already is. That may be because we all have good immune function, and what happens when a person develops cancer, what is the immune reaction to it, and how does immunity play a role in cancers. So, cancers learn to avoid immune destruction, and To enable all these things to happen, you have the cancers have genomic instability. They are fundamental to a cancer cell, and they also have inflammatory cells that are drawn into the tumor. So, they also play a role in allowing all these changes to happen within a cancer cell. Right. So, we will move on to the most important of these all, the first and foremost ability for self-sufficient growth signaling. Now, before we actually go into the details of this, the ability of the cancer cell, we will just talk a little about how a normal cell grows or proliferates. Now, this is a picture that shows you the plasma membrane surrounding the cytosol of a cell, and how is it stimulated to grow or proliferate. Now, this happens due to a series of events that takes place within the cell. The first is that there is a growth factor that comes and attaches itself to a growth factor receptor. The what you see in red spanning the plasma cell membrane is the one that is the receptor, and what like a knob that is sitting on it is the growth factor. So, the first event that happens is the growth factor sits on the growth factor receptor and activates it. Once it is activated, there are a series of events that takes place within the cytosol, the signal transduction, which is mediated by a whole lot of proteins, the RAS being one of the important signal transducer, which ultimately deliver, after a cascade of events, ultimately deliver certain signals to the nucleus, where transcription is activated. Now, once transcription is activated through transcription factors, a whole lot of proteins are generated that are necessary for building new cells, such as proteins that form organelles, proteins that are involved in various cellular activities, metabolism, organelles, and so on and so forth. So, the ultimate signal is delivered to the nucleus. So, there is a growth factor that sits on a growth factor receptor, which induces signal transduction due to various proteins. There is a cascade of reactions that takes place within the cytosol. Ultimately, it goes into the nucleus, wherein transcription factors are activated, and they promote the formation or the transcription of various genes that are necessary for building cellular organelles, and finally, the cell goes into a cell cycle, because cyclin dependent kinases and all those proteins that are necessary are transcribed. Okay, so now what happens is each of these are coded by genes, the growth factors, the growth factor receptors, the RAS, these all have certain genes that code for them. So, when a cancer cell forms, there is a gain of function mutation that converts these normal proto-oncogenes to oncogenes. So, there is a mutation that converts all these, whatever this is, a growth factor or a receptor or a RAS gene or a transcription factor, from its normal proto-oncogene state to a oncogene. So, let us discuss each of this in a little detail. First, we will go to growth factors. Now, growth factors are normally secreted by cells. They need the growth factors for cellular proliferation, but usually the scenario in a normal cell is, if a cell secretes a growth factor, it does not have the receptor for it. The receptor for it will be in a neighboring cell. Now, this is how a certain regulation is maintained, so that the cell does not sort of become overpowerful in itself. It is secreting its own growth factor, which acts on itself to promote its growth. So, the growth factor is secreted by a cell and the receptors are normally present in the neighboring cells. But what happens in cancer is the cell acquires an ability, the cancer cell, to secrete its own growth factor, and it will also have its own receptor. So, this is kind of an autocrine function that happens within a cancer cell. Examples are gliomas, which are special types of cancers that occur in the CNS. They secrete certain growth factors called PDGF or platelet derived growth factor, and also express the receptor for that. And then you also have sarcomas or the mesenchymal tumors that express transforming growth factor alpha and may also express the receptors for the growth factor. So, this is one mechanism by which there is self-sufficiency in growth signal. The second mechanism is with something to do with the growth factor receptors. We saw in the diagram that the growth factors comes and attaches itself to the growth factor receptor. Now, if there is a mutation in the gene that encodes these growth factor receptor, it could convert this receptor into a self-sufficient kind of a protein, in the sense that it will acquire an intrinsic tyrosine kinase activity. So, it may need very little growth factor, even a very minute quantity will enable it to be constantly activated, or it may even do away with the growth factor and acquire the ability to self-activate itself. So, these are various ways in which the growth factor receptors can get activated. So, they can function as oncoproteins when mutated or overexpressed. So, there are examples where growth factor receptors are mutated, such as ERB1 or EGFR in lung cancer, and HER2 is overexpressed in breast cancer. Now, the knowledge of this is very important because we now have drugs that can target these growth factor receptors. For example, EGFR mutations are known in adenocarcinomas of the lung, and today we have drugs against the EGFR receptor, and that can be used in cases of adenocarcinoma of the lung to treat the patients. And of course, we also know about the HER2 gene that may be overexpressed in breast cancers, and we have drugs that can target these the HER2 protein and keep the cancer cells under control. So, the knowledge of these mutations and the altered products is beneficial because it can help us design better drugs for these cancers. The third pathway in which the cell can become self-sufficient is by having an increased amount of signal transducing proteins. Now, one of the most important examples of this is the RAS oncogene, because it is the most commonly mutated oncogene in tumors. Now, normally the RAS has a function where it flips between an active state and an inactive state. So, it is normally bound to a protein called GDP, and it is bound by this protein and it remains in a quiescent state. When it is activated, the GDP converts itself to GTP and the RAS becomes an active, RAS is released and it becomes active. Now, in a normal cell, this is kept under control because the RAS protein has an intrinsic GTPase activity. In other words, as soon as it is activated, its GTPase activity also becomes activated. So, the GTPase activity converts the GTP back into GDP and it binds the RAS again. So, there is a self-contained kind of inhibition in itself. So, the activated RAS, the normal RAS will go back to the quiescent form in a normal cell. But what happens is, in cancer cells, this RAS is continuously activated because it is mutated and it starts to send signals downstream, and finally that goes into the nucleus. Now, what are these mutations? These mutations usually interfere with the breakdown of GTP that I already talked about, or it could lead to the loss of function mutations in what are known as gaps or GTPase activating proteins. Now, the gaps are nothing but enzymes that activate the GTPase. So, they enhance the breakdown of RAS and makes it into a quiescent state. So, in other words, they normally act as tumor suppressors. Now, you could have mutations that interfere with the breakdown of GTP or loss of function mutations in gaps. So, there are lesser number of gaps and that leads to an increased RAS activity. Now, another example apart from RAS is that of the ABL protein. We already saw in one of the previous lectures that you could have translocation of BCR ABL gene in CML, chronic myelogenous leukemia. Now, ABL also acts as a downstream sort of signal transducer, and it can cause increased activation finally within the nuclear transcription factors. So, RAS and ABL are examples of proteins that can cause signal transduction. Finally, for a cell to proliferate, the signal has to go to the nucleus. So, that is the end point of all the signals that reach ultimately allowing the cell to proliferate. So, within the nucleus there are various factors or genes if you may, which are code for transcription factors such as the MYC, MYB, JUN, FOS transcription factors that ultimately in turn regulate cyclin dependent kinases. Now, let me just show you this picture. For a cell to go into cycle, cell cycle, there is a whole lot of events that take place as you can see in this cell cycle diagram. So, it has to go into the synthetic phase, the S phase, and then into the G2 phase, into the mitotic phase and G1 phase. Now, there are two break points in this cell cycle. One is the G1 to S phase and the second is the G2 to M phase, where the cell pauses for a while to see if there is any cell, there is any DNA damage or whatever, and in some instances the cell cycle is stopped because if it is if there is some damage and the cell is not, it is not in the interest of the cell to further proliferate, then it stops there. But these two are the checkpoints wherein a whole lot of regulation can happen. So, you have cyclins that promote the cell to go into the cell cycle, and these cyclins actually bind to what are known as cyclin dependent kinases. So, you have the CDKs binding to the cyclins which drive the cell cycle, and you also have a regulatory mechanism to this, what are known as CDK inhibitors or the CDKIs. So, in a cell that transforms into a cancer cell, you could have gain of function mutations involving the CDK4 or the cyclins, which could drive the cell into increased proliferation, or you could have the reverse, which is the loss of function mutations that involve the CDKIs or the CDK cyclin dependent kinase inhibitors. So, either of these will result in the cell going into an increased proliferative state. Now, having said all this, we should remember that with all these mutations, still the cell needs that the genes that regulate cell senescence and cell death must be disabled for all these mutations to effectively convert a normal cell into a malignant cell. Okay, in summary, we have today in this lecture listed the hallmarks of cancer, the 8 hallmarks, and in some detail we have talked about the self-sufficient growth signaling of cancer cells, and we have discussed that oncoproteins may promote uncontrolled cell proliferation either by the growth factors become independent stimuli for the cells. the mutations in growth factor receptors causing increased tyrosine kinase activity. We saw examples of EGFR and HER2. There could be amplifications of receptors, that is HER2, in breast cancer. There could be mutations of signal transduction genes, example the RAS gene, which is mutated in a whole lot of cancers. We can have overproduction of transcription factors. We saw the MYC gene can be, the MYC protein can be produced in a big number. When there is a translocation, we saw the example of Burkitt lymphoma, when it gets translocated next to an immunoglobulin gene. And finally, we may have mutations that activate cyclins, which drive the cell into an increased proliferation, or we may have mutations that inactivate the cyclin dependent kinases. Both of these actions will cause an increased cellular proliferation. So, all these changes can ultimately lead to the transformation of a benign cell into a malignant one. Thank you.